Are you good? We're good. Hey, look at that. So welcome everybody to GTC. Hopefully you all had a chance to take a look at the keynote that we just had. I think it's going to be a really exciting couple of days that we're going to have here. So thanks very much for coming. I'm excited about the sessions I'm going to be hosting today. I just need to introduce our speaker and then we'll get going. Tom Dean is a full-time research scientist at Google in Mountain View, California. From 1993 to 2007, he was a professor of computer science and cognitive and linguistic sciences at Brown University. He received his Bachelor's of Arts in mathematics from Virginia Polytechnic Institute and State University in 1982, and his Master's of Science and PhD in computer science from Yale in 1984 and 1986. His research interests include automated planning and control, computational biology, machine learning, neural modeling, probabilistic interference, robotics, and spatial and temporal reasoning. And hopefully he has time for some fun on the side in addition to all of that. So please give a warm welcome to Tom. Thank you.
Thanks. Mic on? Yes. Hello and welcome. As Chris said, I'm a researcher at Google. And I'm collaborating with Jim Mutch, who works in Tommy Paggio's lab at MIT. For the last 60 years, the research in biological vision, neuroscience, and the cognitive sciences have more or less paralleled the work in computer vision. And often they shared insights, ideas, and more recently, technology. I'm going to talk about some of the basic principles behind biological vision, which are also principles that have been adapted and adopted by computer vision, and how these can be realized quite efficiently using GPU technology. So from the early work of Hubel and Wiesel back in the 1960s, it became clear that the striate cortex could be characterized, at least in part, in terms of a set of filters that were localized in space and time, that were oriented, and that were bandpass, so tuned to frequencies and scales. Gabor functions and Haar wavelets are typical examples of these. In addition, it was pretty clear that the human or the primate brain in general didn't attend to everything simultaneously and used a variety of mechanisms in order to attend to specific stimuli, particular stimuli that corresponded to a local maxima in some sort of a differential operator. Harris corners, Laplacian operators, they're all examples of that sort of thing. There are lots of other kinds of maps that the brain used, and the most common that was characterized in the early work of Hubel and Wiesel were retinotomic maps that preserved the geometry on the retina in the striate cortex and beyond. But in the decades since, we've found upwards of 30 additional maps that preserve various characteristics of the image and are usually referred to as topographic maps. I'll give you a particular example of a technology that has been used quite a bit in computer vision in the last 7 or 8 years. And it's usually referred to as a bag of visual words as an analog of a bag of words, which is the basic technology that's used in most web searches. So this typical sequence of events in computing a bag of words is you start with some set of coordinates for a set of points. Those could be selected randomly. They could be used, generated from a regular grid, but more often than not, they correspond to the extrema or the interest points generated by an appropriate saliency operator. Given the coordinates, then you can compute feature vectors. And these descriptors correspond to some computation of a local region about the coordinates of the interest points. Histograms of gradients of local patches are the typical thing that's used. The next thing you want to do is you want to reduce the dimensionality in some way, and typically this is done by some method of vector quantization. The easiest way, and the way that's used most commonly and works quite well, is to use k-means to cluster the descriptors, and then just use the indices of the cluster coordinates, or centroids. And then you throw away all of the information that you had about the locations and coordinates, and you summarize the entire image in terms of a vector where the ith component of the vector corresponds to the number of times the code word from the vector quantization has appeared in the image. Now, almost all of these steps can be accelerated using GPUs. And so, in particular, often you resize the image to a standard form. You generate a scale space pyramid, perform some convolutions and smoothing. You apply a differential operator. You find the extrema in the local scale space. The next one you can do on GPU, but often out-of-order CPUs are better for that. You orient the interest point in its frame, and by doing so, you gain some degree of orientation invariance. And then, again, typically, you perform some kind of a decomposition, and Gabor wavelets are a typical example. Compression also can be done fast on a GPU, but I've left that out. Most of the operations that are starred here are ones that can be easily done by using standard packages such as BLAS or FFT that have been accelerated to run under CUDA. In the last 6 or 7 years, there's been a more careful look at some of the neuroscience that proceeded in the previous couple of decades. And one of the most important things has to do with generalizations of contrast normalization. In the neuroscience community, this is usually referred to as divisive normalization. And luminance gain control in the retina is perhaps the most typical of these. But now, generalized versions of this kind of normalization apparently can be applied in various kinds of hierarchical models, the sort of things that we'll be looking at in a moment. Also, it's clear that though in much of the early work we represented the image in terms of the responses of a set of linear filters, clearly the brain is highly nonlinear. And eventually it was clear that there were deficits in using the linear transformations only. And so what people did was they applied some form of a nonlinear transfer function to the outputs of the filters, and typically that was something that saturated the input and then cut off the DC components. So thresholding and half-wave rectification are typical examples of that. And finally, starting with the work of Olshausen and Field in developing various methods of sparse coding that go back to the early work of Barlow in the 50s, we discovered that sparsity in the sense of computing features, most of whose components are zeros, turns out to be not only efficient, but very useful for creating representations that are stable in the sense that small perturbations in the images do not change what the output of the representation is. So again, we'll dive down a little bit deeper into sparse coding because we're going to be talking about that later on. So the objective function for sparse coding is typically characterized in terms of two primary components. One which is a measure of reconstruction error, the first term, and a second which corresponds to a penalty function which encourages sparsity in the outputs. And that can take a number of forms. The most common is to use an L1 regularization. So in this case, the reconstruction error is the difference between x, the input, which is typically a set of patches that are represented in a vector where the columns are the flattened patches, and a coefficient matrix A multiplied times the basis vectors B. So x is now represented as a linear combination of coefficients of the basis b. There's a variety of ways of solving for both the basis and the coefficients. One of the standard ways is to break the problem down into two separate optimizations, each of which are convex, and then to alternate back and forth between the two. And this generally results in a relatively fast convergence to a solution which is quite useful in practice. So in the analysis step, we solve for the basis vector holding the coefficients constant, and our objective function in that case is just the reconstruction error with some constraints on the basis weights. In the alternating synthesis step, we solve for the coefficients holding the basis constant. This one's a little bit more complicated because in this case, the penalty function is a L1 regularizer, and hence the function itself is difficult to compute. However, there are a collection of different algorithms out there now that have been used to do coordinate descent, because coordinate descent not only has an important application in computer vision, but also in terms of compression. So this is a piece of code written in the M language of MATLAB. I doubt that you can read it from where you're sitting, but you can see that it's relatively short and relatively simple. And the nice thing about MATLAB in general is it very closely reflects the underlying mathematics, in particular the linear algebra that describes the algorithm. This will run in straight MATLAB as written, but we use it in conjunction with a framework that was developed by the company Accelerise, and the framework is called Jacket. And Jacket allows us to use this particular piece of code as is, with the only change that we cast the input variables, the a and the y in this case, to be of a particular type, a type that Jacket can run. And as a consequence of this, the algorithm can exploit parallelism in a variety of ways. The most obvious is by using BLAS and some clever programming by the folks who built Jacket. It can exploit data parallelism through matrix multiplication. It can also perform parallel coordinate descent by the use of element-wise operators. So the programmer who thinks in terms of vectorizing his or her code can accelerate things very quickly. Easily, you can get a 20 to 30-fold speed up by just taking your raw MATLAB code and casting it in terms of Jacket types. You can also do some additional optimizations if you move a little ways from standard MATLAB. For example, the line search equations in the inner loop can also be accelerated by some particular constructs that Jacket makes available. The algorithm itself can have many different realizations, and David Mumford introduced a general framework which he referred to as the analysis synthesis framework and its corresponding network, which was a neural network in some sense. It characterized in a rather abstract terms the kinds of networks that we expected existed in the early visual pathways, in particular the striate cortex. And here again, you see the same terms appearing, the residual error or the reconstruction error, and is involved in a loop in which a set of coefficients S are converged by a process which is akin to gradient descent to find the optimal parameters. These kinds of neural networks have been around for a long time, and in fact, the very early work of Fukushima on what was called the cognitron, and then later the neocognitron, are early examples of this. These multi-layer networks were much in vogue in the early 80s, but they ran into some problems, despite the fact that they closely aligned what we knew about the striate cortex, and in particular, the alternation of simple and complex cells in the striate cortex, what Ubel and Wiesel had identified in their research. It was very difficult to solve for the coefficients or the weights in these models. In particular, the idea of propagating error back from the recognition layer to the input layer was made difficult, primarily because no one actually knew how to perform the gradient updates. That changed in the next couple of decades, where first we learned about backpropagation, which enabled us to propagate the weights back, and then some work by Jan LeCun and his group on what were called convolutional nets, which allowed us to exploit certain spatial structure in the networks. And for multi-layer networks plus backpropagation, we finally had a solution to a particular subset of the kinds of problems that Fukushima was looking at. The next insight came from Jeff Hinton and his group at Toronto, and that was that you could train these networks in an unsupervised manner by training the first lowest layer closest to the input, learning the weights for that layer, and then propagating the input through the learned weights to learn the subsequent layers. And this iterative layer-by-layer method was able to find solutions to problems that before were very difficult to find and pretty much we found by hit or by miss by many random restarts of the standard algorithms. Now, Jeff's original work assumed that the consecutive layers within a hierarchical network were completely connected, so they were complete bipartite graphs. This turned out to be obviously problematic when the networks got large and the inputs got more interesting. But very quickly, people took the earlier work for LeCun on convolutional nets and were able to apply that to these frameworks. And the new networks, the networks that are coming out in the last few years, have been able to exploit both the hierarchical structure, the spatial structure, and in the work that I'm going to present today, the temporal structure of video data. So in the last couple of years, these networks, fueled in part by the interest due to the new work introduced by Jeff Hinton and his students, have started to look at generating large collections of these hierarchical models and trying to understand better how they work. And a couple of the people who have been foremost in this correspond to Jan LeCun's lab again at NYU, Yoshua Benjua's work at Montreal, and some work that's happened at Harvard, led by David Cox and some students of Jim DeCarlo's at MIT. Both David and Nicholas Pinto are giving talks here at the conference, and I encourage you to take a look at their posters and their work. The basic idea is relatively simple. You have a parameterized family of models. You generate thousands of these models. You can just use random weights, and that turns out to work quite well. Or you can perform some kind of unsupervised learning in order to update the weights. You test all the models on a screening test, and this is an analog of the idea of high-throughput screening in bioinformatics. And then you choose the best models. And the remarkable thing is that if you generate enough of these models, you get a relatively nice distribution. And if you take models in the long tail, those turn out. Now, these are models that have just performed very well on the screening test, and then you apply them to a test or a set of data that you're really interested in. They often perform extraordinarily well, and this has been a real eye-opener. And the work of Bengio, the work of LeCun have replicated this kind of ability of these networks to perform on a par or better than the state-of-the-art algorithms. So the Bengio work used a GPU framework called Theano that used as its underlying scripting language Python. And the work of Pinto et al. Used another framework called PyCUDA, which is also a Python-based or Python scripting language with metaprogramming capability for programming GPUs. I'm going to focus on a model, a framework that was developed by Jim Mutch and his colleagues at MIT. And it's a framework specifically designed for the kinds of models that I've been describing. These get their inspiration from the cortex, and since they're referred to as cortically organized networks, and CNS corresponds to cortically organized network simulator. So, like the Accelerise product, Jacket, CNS uses MATLAB and its M language for the scripting language. And then it uses C++ in order to describe the kernels in the CUDA sense that will be used to simulate the behavior of individual cells. So the basic model itself is represented as a MATLAB structure that defines the number and type of layers, their dimensionality, the connectivity between layers, and connectivity can span multiple layers in a stack of such layers, the initial value of layer-specific variables, and the like. The only real code that you write that's ultimately going to be used in the simulation is that which is executed by a single cell. And CNS provides a number of macros that make it really easy to refer to and read the cells in different layers. And to find those cells in the sense of what's usually referred to in neuroscience as the receptive field of a cell. That is to say, a cell has a set of other cells that it receives input from, and those are called its receptive field. Finding those in a set of different layers, different matrices with different coordinate systems is often quite difficult, and CNS facilitates this. So here's a simple example of how we would program a network in CNS. So this is a 4-layer network. It has an input layer, which essentially is a buffer for storing an image. It has a layer that has one or more scales, so a scaled version of the input. It has a filter layer in which we essentially apply a set of kernels, convolution kernels, to the scaled input to obtain a 3-dimensional object, which is the 2 dimensions corresponding to x and y, and the 3rd dimension corresponding to the filter index. We then pool that input in terms of the spatial coordinates and often across scales. And so in this case, we're pooling against 2 scales and across the x and y of the filter responses, which are reduced in dimensionality because of the pooling. So here's the basic way that you would define and run a CNS model. This would be a complete model, and it pretty much fits on a single page. This simple model only has 3 layers, the input layer with its corresponding buffer, a scale layer that consists of a single scale where the original image is downsampled by a factor of 2, and then a filter layer on which the filters are Gabor filters, and there are 4 of them, 2 quadrature pairs, and the corresponding coefficients are shown. I'll point out the f params, which is a variable that would be introduced as part of the type of a filter cell, and it will have not only the parameters that will be used to define it, but also the values or weights that will be generated. And those weights will actually be generated in MATLAB and then used within the model, which will be run on the GPU. To run it, you simply initialize it, read an image to get some data. You can then set the values in a given layer by, again, using a simple object-oriented interface, and then you run it. When you initialize it, basically you're initializing the data that's going to be stored on the GPU, and you're allocating the resource corresponding to the GPU. When you've run it, you might have invoked a variety of different complicated steps. In the case of, say, a convolutional net, you might be doing a forward projection, a backward phase. You might be doing some additional gradient descent learning and the like. And all of these things are quite easy to specify within CNS. Here, we're simply doing a feed-forward pass, and then we can extract the output from the third layer, which is the output layer in the case of this simple model. And then when we're done, basically, we tell CNS that's the case, and it frees up the memory and makes the GPU available for other processes. In order to completely specify a cell, and therefore, in addition to have characterized the multiple layers of the network, define the computation associated with a cell within a given layer, we have an object-oriented language that allows us to specify a set of fields. The only thing here that I want to point out is the function fields, which specifies the f-vals. Those are basically going to be the weights of our Gabor filters. And there's one thing to note here is that we specify the dimensions. We're going to give them names, and those names are going to be parts of the macros that will ultimately be generated and be made available when we write code for the kernels. But the DIMs essentially allocate the dimensions of an n-dimensional space or an n-dimensional matrix to the 2 dimensions of texture memory. And we'll get back to that in a moment, but I just wanted to point it out here. For the initialization of the layers, you'll notice that there's a piece of MATLAB code that's called generate Gabor. That's just run in regular MATLAB, and it's going to compute the weights for the Gabor filters and then to normalize them. So a lot of the initialization phase is done in MATLAB. This is all prologue, though, to creating all the parameters that will be needed in the network, or at least the initial parameters. The GPU, or when you're running the GPU model, you might also learn the weights or train the weights of some of the parameters in the models as well. So here's the computational side of a type, of the filter type. And so on the left, you see the code for what essentially is a dot product with a little bit of normalization. And it looks like a simple CUDA kernel code. And the only real difference is that in caps, you'll see a set of functions. These correspond to the macros that expand into the code that it's going to ultimately run. And for example, the first couple of macros that you see there, get layer, y, rf, near, essentially what that makes it easy for the programmer to do is to say, I want you to find a certain number of cells that are within some radius of the current cell. Every kernel is with respect to a particular cell in a given layer. So implicit in all of this is the location of that cell. And so the programmer really wants to know, find me all of the cells in my receptive field, whether that receptive field is in the immediate earlier inferior layer or perhaps at multiple layers. For example, if the receptive field spans multiple scales. On the right is basically the same code with one of of CNS's optimizations where we unroll the inner loops. There are a number of optimizations that CNS makes available that makes it really simple for the programmer to get extra performance out. I mentioned that CNS simplifies the way in which we get the cells, the values of the cells in the receptive field of a given cell in a layer. This is done using a common coordinate system in which the integer indices of the cells within a layer are mapped to a real valued space, a real valued two-dimensional space, which facilitates our ability to get the cells in receptive fields. So that could be all the cells that are nearest to me in a particular layer, layer 1, for example, or all the layers that are within some particular fraction of a unit or a unit in another layer. The idea that everything is mapped to texture memory, the 2 dimensions of texture memory, this really isn't a limitation for most of us for thinking in terms of models that are meant to characterize what's going on in cortex, since the cortex is basically a sheet, and even though there are multiple layers within the sheet, the smallest, the primary component that we often think about when we're building cortical models is something called a cortical column, which is basically a vertical slice through cortex. And so on the right here, you sort of see a graphical rendition of a portion of striate cortex showing the way that orientations and ocular dominance is mapped to the 2 dimensions of cortex. So my real interest in teaming with Jim was to see if we could extend CNS to handle video. I work at video and content analysis at Google, and as you might guess, moving from single images to multiple frames of images in video, thousands of frames, typical, is a considerable challenge. In one sense, you'd think it was easier because a lot of the frames are very similar, but the very fact that they are similar or the small differences that they exhibit provide deep insights into both the kind of properties of still images that you can extract once you have video information, the temporal coherence within the video, and motion, which is in fact what people are most tuned to in the physical environment. So the model basically that Jim and I have been working on takes as input a video. It then breaks it up into smaller stacks. Those stacks are usually contiguous frames of the video. And then the different stacks can be either offset by one frame or multiple frames. That's called the temporal stride. And the height of the stack is called the temporal span. All of the cells within these models have not only a spatial, an X and a Y spatial component, but they also have a temporal component. They may also have a depth component. That depth could be the channels of a color representation, or they could be the responses of a variety of filters at different levels. As each frame comes in, or each stack is placed on the GPU, and a set of 4-dimensional filters corresponding to the 3 dimensions of space and time, plus the 4th dimension corresponding to the filters. And these result in a 3-dimensional object, which again is the 2 dimensions of space and then the filter dimension. And effectively, we build stacks of these within a given layer. So just like the first layer is a stack of individual frames, the second layer, or the output of the convolutional layer, is a stack of 3-dimensional objects that correspond to these richer encodings of the image. Pooling, in this case, can be over multiple scales and multiple times, as is appropriate. So here's a CNS model that captures time. And this is defined in a package of operators and additional functions called a CNS model. So this has an input layer, which again is a buffer for your inputs. But in this case, the buffer can store multiple frames. The multiple frames have to span at least the temporal span of the receptive field of the immediate superior layer. But typically, we put more on the GPU at one time, so the buffer contains enough for many different stacks. And this is a simple optimization that can be tuned depending upon the memory available. The next layer is a normalization layer. It's going to normalize much as it did on a static image, but now it normalizes with a temporal span, in this case, a temporal span of 3. The convolution layer, again, similar to the convolution layers that we saw earlier, but in this case, all these have an extra dimension of time. And then the pooling step in this case is a max pooling step, which is similar in respects to the pooling steps that we saw earlier, except again, it has a temporal component. So to illustrate some of the power of CNS for handling these, I'm going to briefly say something about how we handle spatiotemporal coding. The algorithm that I described before, despite the fact that it runs quite fast in Jacket, it still would take a long time if we had relatively high definition input and we wanted to compute the coefficients for all of the windows of, say, 11 by 11 in a given image. But now, not only are we coding for the frequencies and different orientations, but now we have the velocities and accelerations in the motion features that we would like to extract. And these are obviously key if we want to capture the things that people are most interested in. So running an iterative solver on this would be totally impractical for our purposes. So instead of solving for the sparse coefficients, we learn a good predictive approximation using a technique. Well, actually, there's several techniques, but currently we're using some work that came out of LeCun's lab called predictive sparse decomposition. And the idea is relatively simple. Earlier, I showed you the objective function for sparse coding. Here it's shown again in a simplified form, reconstruction error plus some penalty to encourage sparsity. We're going to add a new function which we want to learn. That function basically is an approximation. It is going to be a function that's going to learn how to approximate the sparse coefficients that were computed by a coordinate descent algorithm or some other optimization algorithm. So the amended sparse coding algorithm is, again, just the two components of the sparse coding objective plus a third, which basically shows the error with respect to the A, which are the sparse coding coefficients, the ones that are computed optimally, and the output of our predictive function for doing sparse decomposition. And we can quite easily learn both the bases, the original bases, and the sparse coding function using a simple gradient descent method to obtain the parameters for both. And then for all intents and purposes, we throw away B, the basis vectors. We're not going to use that. And we use the matrix M, which is a linear combination of the input data with a bias, moved through a nonlinearity, and then adjusted with a gain G in order to do our predictions. Even now, it's still expensive. Instead of doing a complicated iterative algorithm, we're basically doing a couple of matrix multiplications, or in our case, a series of convolutions. but given that there are typically thousands of basis vectors and our corresponding matrix M that allows us to do this quick approximation has a similar dimensionality, the resulting computations are impractical. CNS allows us to use multiple GPUs asynchronously. And we could use it in this case, but in fact we have so many other additional needs for our computations that we do something a little different. We take another trick from the book of biology, and we use a measure of saliency in order to compute the filters that we need. The idea is fairly simple. We're going to start by taking a stack of images from the original video. We're going to run a particular filter over those. It's like a Harris corner, if you're familiar with that, but it's a Harris corner that works in time and that captures an interesting subclass of periodic motion. The output of that is essentially a filter response, a single layer. So here you see the code as it would be written in MATLAB. You'll note that it does a 2D Gaussian smoothing of each frame, and then it uses a pair of Gabor filters in order to compute what's essentially a Laplacian in the temporal domain. This is work by Piotr Dollar, and has been featured in some of the other work that we've done previously. You can realize this as a kernel and for a single layer. And here again you see the code where we're able to access the cells in the input layer for a given cell, the cell defined by this particular kernel operator. We read the layers of that from the values from that layer, and we also read the coefficients for the Gaussian smoother. And then in the same time, as we go through each frame in the receptive field, we apply the Gabor filters in order to obtain the necessary result. Now, anybody who knows a good deal about computer vision knows that if you have a filter, which is the one that we're showing here, which is separable, you can achieve a great deal of performance gain by taking advantage of that. Here we've combined them, so we're going to be duplicating effort, repeatedly smoothing the same frame over and over again, at least as many times as the temporal span of our receptive fields. The nice thing about CNS is it makes it very easy for us to divide this one layer into two layers, one that does the spatial smoothing, and a second that runs the Laplacian in time, and thereby gaining the advantage of the filter being separable. The next step we do is we take the output of the filter responses and we find in a given region of interest the maximum of the local maxima. So not all locations are going to be coded for. Not all locations are appropriate to be coded for. Many of them don't capture any kinds of interesting motion, and the response detector of Dollar allows us to hone in on those response areas or the coordinates of those responses that are most interesting from a human motion perspective. So we find that local maxima, if any maxima exists within each region, and the kernel for doing that is a very simple winner-take-all kernel that operates over in this case, 5x5 windows for our competition layer. In this case, what we may find a max in only a small fraction of the total number of windows, but that's fine. We're not performing much computation in order to do that, so even though we're doing this in a dense way over the entire image, the computations run very quickly. In the final step, we take just the locations that have been designated for each of the regions of interest, and we perform our predictive sparse coding on those locations. In this case, the algorithm is very similar to the dot product or the convolution that we showed earlier. One thing to take note is that even though we've reduced the dimensionality considerably, it may be the case, and in fact, in practice, it is the case, that only on average about 25% of the regions of interest contain local maxima that are of any interest to you. So in 25% of those locations, we're going to code, and in the other 75%, the conditional that's shown here will return immediately. And for those of you who think in terms of warps and what happens to conditional branches within warps, you can imagine that we're doing more work than we necessarily have to do. But because we've reduced the dimensionality considerably, we still get a great deal of performance gain. Using this algorithm, we can get currently about 3 times real time, so about 90 to 100 frames per second in the experiments that are playing with on the data that we've been using. So in summary, I've been talking about biological vision and algorithms that simulate biological vision, but I've taken the opportunity of being here at this conference to focus on the framework that Jim's been developing, and so I'm going to conclude with some of the advantages of CNS. This is still exploratory research, so the ability to quickly write code, modify the code, and build new models is absolutely critical. And on top of that, running on the kinds of data sets that we have at Google is very expensive, and so even doing small-scale experiments could take a long time if it weren't for the case that we're able to leverage GPUs. CNS makes it really easy to build these models, modify them quickly, and then have them run on GPUs automatically without any modification. In that sense, it's very similar to what Jacket affords for the M language of MATLAB. CNS accomplishes this feat because in CNS everything's parametrically defined, except the code, the C++ code that defines the kernel for a particular cell. And even that cell, even that code only communicates with its environment via macros, which simplify the programmer's job to no end. When you compile for a CPU, the kernel macros expand into code that accesses the structures in host memory. And when you compile for a GPU, those same macros expand so that they access GPU memory. I know from experience that CNS shields the programmer from all the details of the GPU. It takes care of thread management. It performs a restricted class of models and computation, but they seem to be ideally suited and easily extended to handle a wide variety of biological models. The memory management helps or assists by selecting the class of memory, whether it's going to be global, texture, constant, etc. It explicitly initiates all the operations that move data back and forth between the host and the GPU. It handles memory alignment. It also handles texture packing because we have these each layer constitutes a large and complicated matrix, an n-dimensional matrix. It maps these onto the two-dimensional texture mapper and then allocates it in appropriate ways so that we can optimize that data. And then finally, as I said, this supports an interesting and, as we keep exploring, growing class of biological models that allow us to deal in particular with things like neighborhood-preserving maps and the abstractions, as we found, cleanly generalize to handle space, time, and scale. And that's it for my talk. Jim and I will be around for the rest of the conference, and we can have a longer discussion with anybody over lunch or outside. and if there's one question or so, if that's okay with Chris, I can take a question.
Yes, thank you, Peter. Does anybody have a question for Tom? I'm sorry.